Imagine trying to build a machine that can build itself. That is essentially the puzzle scientists face when studying how life began on Earth. For decades, researchers have been captivated by the RNA world hypothesis. This idea suggests that billions of years ago, before DNA or proteins existed, RNA molecules served a dual purpose. They stored genetic information like a library archive, and they also acted as enzymes, catalyzing chemical reactions like tiny molecular machines. It is an elegant solution to life's origins. But there has always been one enormous problem for the RNA world to actually work. RNA molecules must be able to copy themselves without any help from protein enzymes. This is the missing piece that has haunted origin of life research for over 25 years. Scientists have found RNA molecules that can copy other RNA sequences. These are called polymerase ribozymes. But here is the catch: every one discovered so far has been large and structurally complex. This creates a frustrating paradox. A self-replicating RNA would need to be big enough and sophisticated enough to catalyze its own copying, but that very size and complexity make it incredibly unlikely to form spontaneously from random chemical reactions. It is like needing a factory to build the first factory. But a remarkable discovery is now challenging this assumption. Researchers from Philip Holliger's group have identified something unexpected. They call it QT45, short for quite tiny 45. This is a polymerase ribozyme made of only 45 nucleotides that is remarkably small, yet despite its tiny size, QT45 can do something extraordinary. It can synthesize both its complementary strand and even a copy of itself using simple building blocks called trinucleotide triphosphates. The discovery suggests that polymerase ribozymes might be far more common in the vast universe of possible RNA sequences than anyone realized. This finding does not solve the entire mystery of life's origins, but it offers something equally valuable. It shows that self-replication might not require molecular machinery as complex as we once thought. Sometimes the most profound answers come in the smallest packages. Let me start with a simple question. What if I told you that RNA, the molecule we usually think of as a simple messenger, can actually act as an enzyme? This is where things get really interesting. A ribozyme is exactly what it sounds like. It is a ribonucleic acid enzyme. In other words, it is an RNA molecule that can catalyze chemical reactions just like protein enzymes do in your body. Right now, a polymerase ribozyme takes this concept even further. Think of it as an RNA molecule that can stitch together individual building blocks called nucleotides to build entirely new RNA strands. In our modern cells, we have protein enzymes called polymerases that replicate our DNA and RNA. They are essential for life as we know it. A polymerase ribozyme does a similar job. But here is the twist: it is made of RNA itself. This matters enormously for something called the RNA world hypothesis. This idea suggests that early life on Earth might have relied entirely on RNA before proteins and DNA evolved. For this hypothesis to work, you need RNA that can copy itself. That is exactly what a polymerase ribozyme does. But there is a catch. RNA has a serious limitation. Compared to proteins, proteins are built from 20 different amino acids, giving them incredible chemical versatility. RNA is built from just 4 nucleotides. This limited toolkit makes it much harder for RNA to perform complex chemistry. That is why finding an RNA molecule that can act as a polymerase is such a big deal. It shows that despite its chemical limitations, RNA can still perform one of the most fundamental tasks of life: copying genetic information. And this brings us to an exciting recent discovery that changes how we think about the origins of life. Imagine trying to build a machine that can build an exact copy of itself. Now imagine that machine is made of a material that tends to fall apart. This is essentially the challenge that scientists face when studying how life might have begun. At the heart of this puzzle lies something called the RNA world hypothesis. The idea is elegant in its simplicity. RNA molecules can do two things: they can store genetic information like a library of blueprints, and they can act as catalysts, speeding up chemical reactions like tiny molecular machines. In theory, a single RNA molecule could both carry instructions and execute them. That combination could enable self-replication, the fundamental process that allows evolution to begin. But here is where things get tricky. For an RNA molecule to work as a catalyst, it needs to fold into complex three-dimensional shapes. Creating those shapes requires a fairly long chain of nucleotides, typically over 100 building blocks. The problem is that longer molecules are exponentially harder to copy accurately. The copying process itself is error-prone, and mistakes accumulate quickly. This creates a frustrating paradox. A polymerase ribozyme needs to be long enough to function as a catalyst. But the longer it gets, the more difficult it becomes to replicate faithfully. It is a classic chicken and egg problem. For over 2 decades, researchers have been stuck in this trap. Scientists have engineered polymerase ribozymes that can copy other RNA sequences with impressive efficiency. But these molecules are massive by molecular standards, far too large and complex to copy themselves. The assumption took hold that sophisticated RNA copying required sophisticated RNA machinery. Meanwhile, smaller ribozymes remained unexplored territory. Most researchers assumed that if you wanted polymerase activity, you simply needed a bigger, more complex molecule. This assumption created a blind spot that would take an unexpected discovery to illuminate. What if the answer was not about building bigger machines but finding smaller ones? Here is the script segment for your video chapter. Imagine trying to write a copy of a book by hand, but the book itself contains the instructions for how to write. This is essentially the challenge that has faced researchers studying the RNA world for years. Scientists have engineered polymerase ribozymes, which are RNA molecules that can build other RNA strands. But there was always a catch: these ribozymes were too large and complex to copy themselves efficiently. It was a classic chicken and egg problem. Now researchers have achieved something remarkable. They have discovered a tiny polymerase ribozyme called QT45 that is only 45 nucleotides long. Let that sink in for a moment. This molecule is small enough to be copied in a reasonable time frame, yet it still functions as a working polymerase. Even more impressive, QT45 can synthesize both its complementary strand and even a copy of itself. The researchers found this little marvel using a technique called in vitro evolution. Think of this as natural selection in a test tube. They started with a pool of random RNA sequences and applied selective pressure to find molecules that could perform polymerase reactions. Through many rounds of selection, they coaxed out sequences that were both compact and active. Why does size matter so much in the primordial world? A massive complex ribozyme would take too long to copy, errors would accumulate, information would be lost. But a compact ribozyme, one that is short and efficient, could potentially sustain a self-replicating system. This discovery fundamentally changes how we view the probability of life emerging from an RNA world. It suggests that functional polymerases might be far more common in the vast space of possible RNA sequences than we ever imagined. The bridge between chemistry and biology might be easier to cross than we thought. So how does this tiny molecular machine actually copy itself? Let me walk you through it, step by step. The process starts with recognition. The QT45 ribozyme finds and binds to a template strand, which could be itself or a complementary copy of its own sequence. Think of it like a lock finding its key. But in this case, the lock is also helping to build a duplicate key. Once bound, the ribozyme gets to work. It uses building blocks called trinucleotide triphosphates, which are essentially three-letter chunks of RNA code. This is actually quite clever. Instead of adding letters one at a time like most polymerases, QT45 works with these three-letter packages. The ribozyme positions these building blocks along the template and catalyzes the formation of chemical bonds between them, specifically phosphodiester bonds, which are the molecular glue that holds RNA strands together. Now, here is where things get interesting. The reaction requires specific conditions to work. The researchers found that it operates best in mildly alkaline conditions within ice, specifically at around -7 degrees Celsius. The ice creates concentrated pockets where the molecules can interact more frequently. When it comes to accuracy, QT45 does a respectable job. It achieves about 94% fidelity per nucleotide, meaning it makes mistakes roughly 6% of the time. For a 45 nucleotide sequence, that error rate is significant, but not catastrophic for a primitive replicator. The biggest limitation right now is efficiency. The yields are quite low, around 0.2% after 72 days. That means out of every 1,000 attempts, only about 2 successful copies are made. The ribozyme also has limited precisivity, meaning it tends to fall off the template before completing the full copy. These limitations highlight that QT45 is not a perfect replicator yet. But remember, this is a molecule that emerged from random sequences in a laboratory. The fact that it works at all is remarkable, and it suggests that similar capabilities might be surprisingly common in the vast landscape of possible RNA sequences. So what does this discovery actually mean for our understanding of life's origins? For decades, the RNA world hypothesis has faced a stubborn problem. The idea that RNA molecules could have served as both genetic material and catalyst makes elegant sense. But there was always a catch. Every polymerase ribozyme we had discovered was too large and complex to have plausibly arisen spontaneously from random chemistry. It created a paradox. The molecule needed to be complex enough to copy itself, yet simple enough to emerge by chance. QT45 breaks this paradox wide open at just 45 nucleotides. It sits comfortably within the realm of molecules that could form spontaneously under the right conditions. This does not prove that life actually started this way, but it provides something almost as valuable. It shows us that self-replication does not require molecular machinery of overwhelming complexity on early Earth. Such a molecule might have found ideal conditions on mineral surfaces, which could concentrate RNA building blocks and provide a scaffold for assembly. Temperature cycles from day to night, or near hydrothermal vents, could have driven strand separation, allowing replication to proceed without the sophisticated enzymes cells use today. Looking forward, synthetic biologists see exciting possibilities. A self-replicating RNA system could become a platform for engineering new biotechnologies, creating molecular machines that grow and evolve on their own. Of course, challenges remain. QT45 works, but it is not yet fast or accurate enough for true exponential amplification. Demonstrating evolution in a fully self-replicating system remains an unmet goal, but let us step back and appreciate what this means. A molecule that can copy itself is essentially a proto-organism with Qt45. We have moved from theoretical possibility to demonstrated reality. We are one step closer to watching chemistry become biology, and perhaps one step closer to understanding our own deepest origins.